Hi, my name is Judah Engel, and I'm an undergraduate at Columbia doing research in the Herbert and Florence Irving Institute for Cancer Dynamics. And today I'll be discussing simulation-based inference methods for cancer modeling. So, first, it's important to discuss a novel single-cell sequencing technology that's been developed at Memorial Sloan Kettering called Direct Library Preparation Plus, which has the potential to inform our understanding of cancer processes. In contrast with bulk sequencing, which looks at more of a general overarching kind of look at the genome of a tumor, single cell DNA sequencing is ideal for studying tumors exhibiting heterogeneity, because bulk sort of kind of mixes all the cells in the tumor together. Single cell DNA sequencing allows us to kind of study like subclonal populations, and also in contrast with things like RNA sequencing, which require pretty advanced inference methods in order to gain understanding of copy number variations. And to the right, you can sort of see a schematic of this wet lab technique that's been developed, which is not the focus of this presentation, but is very interesting and novel nevertheless. So the data that's been collected is in the form of a matrix that with each row corresponding to a different cell in the tumor. We have around 1,500 cells per sample in our study, and each column corresponds to a different bin holding the copy number of a specific 500,000 base pair region of the genome. So you can see a heat map in this bottom right corner. The average ploidy level is normally around 2, indicating a healthy diploid, but these values can really vary depending on the level of mutations in a particular sample. We built a stochastic model to simulate the evolution of ovarian cancer, and the model takes around 76 parameters as input, including known variables such as cell turnover rate and population size, as well as unknown parameters such as mutation rates and selection rates. So these mutations can include things like whole genome duplication, focal amplification, and deletions. And the selection rates are sort of a conditional probability defined by given a certain event occurring, what's the probability of a particular cell passing on its mutation or being destined for apoptosis. The model also produces data in the form of a copy number table, but it also produces a phylogenetic tree detailing the evolutionary history of the cells in the tumor. So this is really key. The idea is that if we can use this stochastic model and fit it properly such that it's able to produce copy number tables that resemble the copy number tables sampled from real tumors, we are able to look back at sort of the mutational events that occurred that gave rise to this copy number, allowing us to see its evolutionary history and in turn allowing us to better understand the progression of this disease. So each of these parameters in the model can be described using a probability distribution, and regularly the posterior distribution is calculated using the prior distribution, which ours is often set to a uniform distribution, which is uninformative. You have a piece of evidence, which would be the sample, and a likelihood function that helps connect the prior to the posterior. However, due to the complexity of our model, we have an intractable likelihood function, which requires us to utilize numeric approach to evaluating the posterior. In particular, we use approximate Bayesian computation. In this scheme, which you can kind of see on the right, we sample from a uniform prior distribution. It doesn't have to be uniform, but in our case it is. And we produce data from the simulator using sampled parameter values from the prior, and we then calculate a summary statistic for those simulated data and calculate a distance between those simulations and the real sample, and we reject all samples that fall below, or all simulations that fall below some similarity threshold, basically. The issue with approximate Bayesian computation, at least the traditional rejection ABC, is that it's heavily influenced by the choice of the summary statistic. In particular, when we utilized Euclidean distance for our summary statistic, we found that the posterior distribution was skewed very much to the left, which kind of seems as if it's favoring little to no mutations at all, which would make sense given our understanding of how Euclidean distance would be minimized. We also we then decided to kind of utilize a variant of approximate Bayesian computation that utilizes random forests, which circumvents the need to explicitly define a summary statistic and instead uses a vector of covariates. It's also more efficient because it doesn't throw away simulations, but rather just weights them all differently to actually calculate this posterior. And we used a structure similar to a site frequency spectrum by averaging the columns of these copy number tables. So here's another figure, and it shows you the posterior centered around this fixed parameter value that we set in just a trial run when we were performing ABC random forest on like a ground truth table developed from the simulator itself with a known parameter value. Now we're currently working on incorporating information on clonal ancestry using a CNN. So compressing the copy number table into a site frequency spectrum reduces spatial information corresponding to clonal ancestry. So in these copy number tables, rows that are close together indicate cells that have closer common ancestors. And so when we kind of average everything out along the column, we're sort of just getting rid of this this spatial information that corresponds to this clonal ancestry. And so utilizing a CNN, which is a neural network that's often used in image processing, we hope to extract spatial information that would allow us to incorporate some of this information on clonal ancestry into the inference model. And after fitting for a single parameter, which was the missegregation rate, we need to expand our approach to allow for the fitting of multiple parameters. This poses challenges related to computational complexity with regards to a very simple algorithm like ABC, because it requires that you sort of simulate every combination of parameters. So we plan on integrating neural network-based approaches that perform well in high-dimensional problems, something like this example, which is generative adversarial training for simulation-based inference, which utilizes like neural networks to perform inference. All right, that's just it for the talk. Thank you, and let me know if you have any questions.